The terms recombinant DNA technology, DNA cloning, molecular cloning, or gene cloning all refer to the same process: the transfer of a DNA fragment of interest from one organism to a self-replicating genetic element such as a bacterial plasmid or cloning vector. The DNA of interest can then be propagated in a foreign host cell. This technology has been around since the 1970s, and it has become a common practice in molecular biology labs today. Cloning is frequently employed to amplify DNA fragments containing genes and essential step in their subsequent analysis. So, how does molecular cloning work? So, cloning of any DNA sequence involves the introduction of a foreign piece of DNA into an extra chromosomal element or clonal vector of an organism, which then produces copies of the vector as it replicates itself, thereby amplifying the DNA of interest. The whole process can be summarized in the following steps. First step is fragmentation. Initially, the DNA of interest needs to be fragmented to provide a relevant DNA segment of suitable size. Preparation of DNA fragments for cloning is frequently achieved by means of PCR, but it may also be accomplished by restriction enzyme, digestion, and sometimes fractionation by gel electrophoresis. Next step is ligation. Subsequently, a ligation product is employed whereby the amplified fragment is inserted into a vector. The vector, which is frequently circular, is linearized by means of restriction enzymes and incubated with the fragment of interest under appropriate conditions with an enzyme called DNA ligase. Third step is transfection. Following ligation, the vector with the insert of interest is transfected into cells. Most commonly, electroporation is employed, although a number of alternative techniques are available, such as chemicals, sensitization of cells. Next step is screening and selection. Finally, the transfected cells are cultural, as the mentioned procedure are of particularly low efficiency. There is need to identify the cell colonies that have been successfully transfected with the vector construct containing the desired insertion sequence. Modern cloning vectors include selectable antibiotic resistance marker, which allow only cells in which the vector has been transfected to grow. Additionally, the cloning vectors may contain color selection markers, which provide blue or white screening on X-gel medium. So, last step is confirmation. Nevertheless, these selection steps do not absolutely guarantee that the DNA insert is present in the cells obtained. Further investigation of the resulting colonies is required to confirm that cloning was successful. This may be accomplished by means of PCR restriction fragment analysis and DNA sequencing.
Now, let's proceed to human cloning. Human cloning is the certain of genetically identical copy of an existing or previous existing human being. It is a form of asexual reproduction, meaning that the offspring created from the cloning process will be the same as the individual that is being cloned. Human cloning is a controversial issue that has been discussed in recent years. There are many ethical issues surrounding its potential use. The supporters of human cloning argue that it could help individuals, like aiding in medical research that could use to treat a variety of medical conditions and diseases, while the opponents of human cloning argue that it's unethical. It could lead to the destruction of human life and the human species. The ethical implications of human cloning are still being debated, but it is clear that the potential benefits of cloning could be great. However, the potential risks of cloning remain unknown and must be considered carefully before any decision is made about the future of this technology.
Ethical concerns about cloning may be broadly divided into two categories. Concern about the effect of cloning on the animal and human welfare. Objection to the principle of cloning to producing an animal by a means other than fertilization. Currently, cloning is associated with an increase in animal suffering when compared with production of animals by standard breeding method. These concerns are somewhat mitigated by the fact that most of these findings are not unique to cloning. They are also associated with other procedures that have been generally accepted as worthwhile, such as in vitro fertilization, embryo production, oocyte transfer, and embryo transfer. Additional worries center on the impact of cloning on the entire animal population, most frequently in relation to the species' genetic diversity. This is a valid worry for some species and applications, such as dairy cattle where a single bull may produce thousands of calves. However, this has less to do with the cloning of a bull itself and more to do with the science of semen freezing and distribution. It seems unlikely that the few companion animals that will likely be cloned will have an impact on the population as a whole. Therefore, remaining concerns about consumption of food from cloned animals is likely based on principle than on actual potential harm. Cloning geldings or castrated horse who have proven to be outstanding competitors is one significant proposed usage, saving genetic types that would otherwise have been lost and potentially increasing genetic variation in horses. On the other hand, concerns about human health focus mainly on consumption of food produced from cloned animals. However, the FDA and the European Food Safety Authority concluded that the consumption of meat or milk from cloned animals possess no public health risks, and this is based on years of study. The question of whether it is morally wrong to clone animals or whether doing so amounts to playing God by making embryos without using fertilization is a crucial ethical conundrum. Despite the fact that cloning is often regarded as an anomaly, similar worries have been expressed with each new method of reproduction that has been developed. The public's moral disgust with cloning has grown as a result of the way it is maybe represented as a bad thing in science fiction, literature, and films. Arguments in opposition to these moral objections claim that cloning already occurs in nature in the form of identical twins, that people have been creating plants and animals through unnatural means since the first time they planted the seed or crossed a cow with chosen bull, and that this is just the latest advancement in the same direction. Prior to the birth of Dolly, embryonic cloning had been practiced for more than 10 years with little to no public attention. Even the announcement of the birth of two lambs cloned from embryonic stem cells a year earlier had no noticeable effect on the public. Thus, it appears that the main moral issue of public concern is not the production of embryos without fertilization, but the production of embryos from cells of an existing known animal.
So now, mag-proceed naman tayo sa mga negative effects ng human cloning, no? So, isang article, may title na Human Cloning Endangers Humanity and Should Be Prohibited by Pimentel. So, dito, since alam naman natin, di ba, na ang mga scientists natin ay patuloy sa pagre-research, patuloy sa pag-experiment, no? And about ng human cloning, kailangan pa rin nating gumawa ng kilos para maban ito. Kasi may mga nag-impose to ng mga legal barriers, no? And yung immortal undertaking. So, ano nga ba yung pinopoint out dito ni Pimentel, no? Sabi niya kasi dito, as means of creating other human beings, which is yung cloning, automatically, ang mangyayari, mai-eliminate yung man from his vital role as the indispensable partner of a woman in the preservation of the human race. So, isa yung makakaapekto yung role ng mga kalalakihan ay mawawalang bisa, no, sa kanilang mga asawa. Lalong-lalo na, sabi dito, it only takes one woman's womb to perpetuate a race of cloned individuals. And syempre magiging complicated yon kapag gano'n ang nangyari. And ano na lang mafe-feel, 'di ba, ng mga lalaki. So sila yung mag-a-adjust, parang gano'n, 'di ba? So hindi lang naman dito sa Pilipinas, no, na i-raise itong kantong concern. Actually, marami pang countries, no. Merong 46 countries, including yung Germany, France, Belgium, Denmark, Holland, Russia, and Japan, no. Ilan lamang ito sa mga nagpo-prohibit ng human cloning in various ways, no. And we believe, no, na syempre ang human embryo, 'di ba? Alam natin that the human embryo pulsates with life of a human being, no? It is a distinct individual. So, with his or her own human characteristics and an immortal soul that set him or her apart from all others. So, yun yung pinaniniwalaan natin eh. Kasi it is God's design sa human life. And also we believe, no, na yung life, it begins at conception. At wala nang adequate reason na nag-i-exist to justify the use of genetic engineering biotechnology that makes life away from an embryo in the guise of scientific advancement or any other alibi. So, before we agree sa human cloning, isipin natin yung mga negative effect nito at kung ano ba talaga yung nature ng tao, kung paano sila nagpaparami, na yun yung design ng Diyos sa atin, kung paano tayo magparami. And pag merong nabago sa mga ways na yon, isipin natin yung malaking impact nito, no, sa pagbabago, lalo na sa mga future generations natin. So, yun lang po yung about dito sa human cloning endangers humanity and it should be prohibited.